Hi, everyone. So I'm going to be talking today about our work called Graph Convolutional Networks for Epigenetic State Prediction using both sequence and 3D genome data. So this is joint work with my advisor, Dr. Yanjun from the University of Virginia. So just a quick preliminary note before I get going. So we use the following words interchangeably: epigenetic state with chromatin profile, and 3D genome with chromosome conformation. So throughout the talk, I'll sort of use those interchangeably. So alongside genes are chromatin elements such as transcription factor binding and histone modifications, represented by these colored shapes above the DNA here. And these are regulatory factors that control whether a gene is expressed or not, making them important to predict and understand. So we define the chromatin profile or the epigenetic state of a DNA window Xi as the chromatin elements that are present at that location. So this includes transcription factors, histone modifications, and accessibility. So epigenetic state prediction is then the process of taking DNA sequence windows as input and outputting the active chromatin elements at those windows using some model F. So given some new DNA windows, the model hopefully will be able to accurately predict the chromatin elements at each of those windows. So previous state-of-the-art methods for this task predict the state of each DNA window independently of all other windows. So they'll take one window, predict the chromatin elements that are present there, move to the next window, predict the chromatin elements, and so on. So these types of models are fairly accurate due to the influence of local sequence features on the chromatin elements. So for example, maybe this red diamond shape might be representative of a transcription factor, and that might bind to window X2 because of this subsequence TATGA. However, some elements such as this blue circle, which might be representative of a different transcription factor, may be present with no sequence features in the window. So one hypothesis for this is due to what's known as chromosome conformation, or the 3D genome, where DNA actually folds in the chromosome. And because of this, windows may be influenced by sequential features which are really far away in the linear space, but really close in the 3D space. So our goal is to incorporate this type of structure into our prediction method so that we can consider these long-range interactions for predicting these chromatin profiles of each DNA window. So this 3D shape of DNA can be represented by high C data, which indicates physical contact between two windows in the genome. And these contacts can be represented as a graph where nodes indicate DNA windows and edges indicate contact between two windows. So this type of 3D structure of the genome can really just be represented by an adjacency matrix that we call A, where again, the nodes are the DNA windows, and a positive cell would indicate that there's some sort of contact between those two windows, which is representative of the 3D shape of the genome. So our proposed method to incorporate this data we call ChromeGCN, which is the first deep learning method to combine both sequence and 3D genome data for chromatin profile prediction. So it's about incorporating this type of 3D structure via high C data into this prediction method to predict things such as transcription factors, histone modifications, and so on. So formally, our goal is, given DNA window Xi, we want to predict the probability of a set of epigenetic labels that we call Y hat sub I, which is just a vector of L total chromatin profile labels. And our method, ChromeGCN, consists of 3 different modules, which I'll go over individually here. So the first module is extracting local sequence features using a convolutional neural network. So this module FCNN maps each local sequence window Xi to a vector that we call HI of dimension D using a shared convolutional neural network. So this is done for each window independently of one another, which for each window, it takes in the sequence and outputs a vector representation that encodes that sequence information. So once we have these vectors H, The second module, which is the main contribution of our work, encodes this 3D conformational relationships between all windows, between all window vectors using a graph neural network. So without going into the details of how this graph neural network works, each of these HI vectors, which represent the sequence features for a particular window, is updated to a new representation that we call ZI using a function of itself and its neighbors indicated by the high C adjacency matrix A. So now this output vector, ZI for each window, encodes both the sequence information and neighboring sequence information that we're using from the high C data. So once we have these Z vectors for each window, the final module just is simply a linear classifier to predict the L epigenetic labels for each of those windows. So sort of putting all this together, our model looks like this, where the first step is extracting local features from the sequence using a convolutional network. The second step is extracting long-range 3D interactions using graph neural networks. And finally, the prediction module to predict the epigenetic labels for each window. So I'll just quickly go over how we try to validate this model. For epigenetic state labels, we use all transcription factor, histone modification, and DNA accessibility data from ENCODE using two commonly used cell lines, GM12878 and K562. And for high C data, we use the interchromosomal contact matrices, which tells us edges within a specific chromosome. So based on this high C type of data that we're using, we split the data by chromosomes using 16 of them as training, 3 as validation, and 3 as testing. So each sample is really one chromosome where we want to predict the labels for all windows within that chromosome. So if you want to use this model to predict on new windows, you really need a full chromosome because we're using all the interactions within that chromosome to predict windows. So we compared 2 commonly used independent window-based deep learning methods, the first 2 here, and we introduced 2 variants of our method, the proposed ChromeGCN model that I talked about, as well as a recurrent neural network version, which really just replaces the graph module with an RNN. So it uses the full linear chain of windows rather than this known 3D structure. So it's just a way to validate whether using the known 3D structure is important, or if we want to just consider all the linear chain of windows. So we find that across both cell lines, incorporating this long-range structure into the prediction model helps improve accuracy when compared to these independent window-based methods. But most importantly, comparing the GCN approach versus the RNN approach, we found that using these known 3D relationships or the known conformational structure of DNA helps the best out of all the models that we tested. So lastly, I'll just talk about a post-processing method that we call high C saliency map. So this is after we've trained the model and want to analyze what the model learned. So while high C maps tell us which pairs of DNA come in contact with each other, they don't tell us which contacts are important for a specific chromatin element to be present. So in other words, it just tells us where the contacts occur, but it doesn't tell us which chromatin elements are affected by those contacts. So we propose what we call high C saliency maps, which is just a method to identify the important 3D relationship of the important contacts for ChromeGCN's predictions. So this is done by taking the gradient of the output y hat for label L with respect to the high C adjacency matrix A. So this will tell you the important edges in this high C graph for a particular label L. So just to show a quick example of what I mean by that, here's an example of our high C saliency map for the transcription factor YY1 in chromosome 8 of cell line GM12878. So what this shows here is the importance of all 500,000 edges that we use for this chromosome, where the darker the line is, the more important that edge was for predicting the ground truth YY1 value. So we can kind of zoom in on particular subspace of this full adjacency matrix and see which windows are actually important for this YY1 transcription factor. So just in conclusion, we propose a new method for incorporating chromatin contact information for this chromatin profile prediction task. And we found that incorporating this type of 3D data helps improve prediction accuracy. And lastly, we propose a new method to find the important high C edges from the high C graph. So our code and data, all the data that we use in the code is available on GitHub. And I just want to thank my advisor, Dr. Chi, and my many collaborators over the past few years.
So thank you, Jack. So I'm checking now the questions. We have a couple of questions. We have one from Peter, who's asking, is the window size fixed? Can windows overlap?
Yeah, so the window size is fixed for our training. We use windows of 1,000 length, and they're non-overlapping, but we can adjust that. I mean, that was just the formulation we used. We actually tried to use the same data processing as the deep C and expecto methods. So, but that it's just an arbitrary choice, and that can actually be changed.